Good morning, everyone. I am Dr. Kalpana Sharma, Associate Professor, Department of Zoology, S.P.C. Govt College, अजमेर। बच्चों, मेरे आज के लेक्चर में मैं आप लोगों को क्रोमोसोम्स के स्ट्रक्चर एंड फंक्शन के बारे में बताने जा रही हूँ। अब देखिए, क्रोमोसोम्स क्या होते हैं। क्रोमोसोम्स आर मोस्टली मेड अप ऑफ डीएनए। काय के बने हुए होते हैं क्रोमोसोम्स? डीएनए के बने हुए होते हैं। और क्रोमोसोम्स कहाँ पे पाए जाते हैं? क्रोमोसोम्स आर फाउंड इन न्यूक्लियस। सबसे पहले क्रोमोसोम्स को हॉफमाइस्टर ने ऑब्जर्व किया था और वाल्डेयर ने क्रोमोसोम नाम दिया था। अब हम देखेंगे मॉर्फोलॉजी ऑफ क्रोमोसोम। क्रोमोसोम किस तरह के होते हैं? द मॉर्फोलॉजी ऑफ क्रोमोसोम्स कैन बी बेस्ट स्टडीड एट द मेटाफेज और एनाफेज ऑफ माइटोसिस व्हेन दे आर प्रेजेंट एज डेफिनिट ऑर्गेनेल्स बीइंग मोस्ट कंडेंस्ड और कॉइल। अब हम देखेंगे नंबर ऑफ क्रोमोसोम्स। द नंबर ऑफ क्रोमोसोम्स in a given species is usually constant, containing diploid number 2n chromosome in the somatic cell and n number of chromosome in the sex cell. For example, in Ascaris it is 2, while in certain protozoan the number is there are more than 300 chromosomes. In Paramecium, 30 to 40 chromosomes. In Radiolarians, as many as 1600. In Hydra vulgaris, 32. Musca domestica, 12. Rana, 26. Columbula livia, 80 chromosomes. Oryctolangus cunicellus or the rabbit has got 44 chromosomes. Gorilla has got 48. And Homo sapiens, man, has got 46 chromosomes. The chromosome number are also helpful for taxonomy. In the angiosperm, the most frequent haploid number is 12, and members of this group have a range from 3 to 16. Similarly, in fungi, haploid number ranges from 3 to 8. In primates, this haploid number is from 16 to 30. This haploid set of chromosome present in the nucleus of gametes. While in a diploid cell, there will be 2 genomes. The diploid cells are the somatic cells in the body, and the diploid cell gets the diploid set of chromosomes by the union of the haploid male and female gamete in the sexual reproduction. Now the size of the chromosomes. Chromosomes ranges on an average from 0.5 to about 30 mu in length and from 0.2 to 3 mu in diameter. The relative number of chromosomes generally differ in the nucleus, but at a time all chromosomes of a cell may be of the same size. Plant cell normally possess larger chromosomes than the animal cell. Now the shape of the chromosome. The shape of the chromosome is changeable from phase to phase. The shape of the chromosome may be rod-like, twisted, spiral, curved, or filamentous, depending upon the position of the centromere, which lies in the primary constriction. Now, on the position of the centromere in a chromosome, the chromosomes are classified as telocentric, acrocentric, submetacentric, and metacentric. As in the diagram, you can see the A denotes the telocentric. Now, this telocentric, what is telocentric chromosome? Telocentric chromosome means in such chromosomes, centromere occupies terminal position so that only one arm is seen. Centromere possesses the terminal position and only one arm is seen. So they are rod shaped. Now next one is acrocentric. In acrocentric type of chromosomes, centromere occupies subterminal position. It is at the centromere at the subterminal position so that one arm is very short and another arm is very long. And another one is submetacentric. In submetacentric type of chromosomes, the centromere is situated slightly away from the middle so that two arms are unequal. They are L-shaped. The last one is the metacentric. In metacentric chromosome, the centromere lies in the middle so that the two arms of the chromosomes are equal, hence they are V-shaped. Now coming to the structure of the chromosome, स्ट्रक्चर देखें तो क्रोमोसोम्स जो है वो किस किस चीज से मिलकर के बनता है? पेलिकल, मैट्रिक्स, क्रोमेटोनिमेटा, सेंट्रोमियर, that is primary constriction, secondary constriction 1 and 2, satellite and telomere. Now in this diagram you can see a structure of a typical somatic chromosome. In this you can see that the chromosome has got the first part, that is pellicle. What is pellicle? Pellicle is the outermost covering. Pellicle is the outermost covering of the chromosome and it is exceptionally thin. Some scientists, however, deny its presence, but photographic evidence supports its presence. The next part is the matrix. As you can see, the filling portion or inside the pellicle there is matrix. The matrix consists of proteins and lipids. किस किस से मिलके बनता है matrix? Proteins and lipids and the chromatonemata. और अंदर जो धागे लाइक संरचनाएं हैं ये है chromatonemata. They are embedded in the matrix. Its exact function is unknown. थर्ड वन इज क्रोमेटोनिमेटा. ये दोनों साइड पे आप जो देख रहे हैं ये है क्रोमेटोनिमेटा. क्रोमेटोनिमेटा, ईच क्रोमोसोम कंटेन टू क्रोमेटोनिमेटा which are usually spirally twisted around each other and lie embedded in the matrix. Each chromatonemata is 800 Armstrong thick along the entire length of the chromatonemata. Are found small chromomeres. क्रोमेटोनिमेटा के अंदर सारे स्मॉल ग्रेन्यूलाई क्या है? क्रोमोमियर्स. Chromomeres in form of dense granule-like structure. The two chromatonemata of a chromosome are joined together at a point known as primary constriction. यहाँ पे आप देख रहे हैं पार्ट इसका प्राइमरी कंस्ट्रक्शन. प्राइमरी कंस्ट्रक्शन को, कॉन्ट्रिक्शन को हम क्या कहते हैं? सेंट्रोमियर भी कहते हैं. ये प्राइमरी कंस्ट्रक्शन वो वाला भाग है जहाँ पे दो क्रोमेटोनिमेटा आपस में आकर के ज्वाइन होते हैं. तो अब हम डिस्कस करेंगे सेंट्रोमियर के बारे में कि सेंट्रोमियर क्रोमोसोम का किस तरह का भाग है. सेंट्रोमियर आर कप लाइक और प्लेट लाइक स्ट्रक्चर, 200 to 250 Armstrong in diameter. It is the most important structure of the chromosome. ये important structure क्यों है? क्योंकि इसके कई सारे फंक्शन हैं. फर्स्ट फंक्शन है, it serves to provide attachment to spindle fiber during cell division. Second, it is also important for chromosomal movement in the cell division. Its location is marked by a constriction in the chromosome known as primary constriction. The structure is centromere is very complicated. सेंट्रोमियर जो है वो तीन चीज़ों से मिलकर के बना होता है. आउटर लेयर, middle layer. Middle layer is a dense and inner zone, and the middle zone helps in the chromosome to maintain relation with the spindle. अगला भाग इसका हम देख रहे हैं secondary constrictions. Secondary constriction जो है, there are two secondary constriction as you can see in the picture of a chromosome. There are two secondary constriction. The secondary constriction number 1 is known as nucleolar organizing region, as it is associated with the formation of nucleolus. And the second constriction is known as secondary constriction number 2, which is known as satellite. You can see in the picture the satellite body. The secondary constriction number 2 is different from the secondary constriction number 1. It represents the site of breakage and subsequent fusion. Now we'll see the next part of the chromosome, that is satellite. Satellite is a small spherical part of a chromosome distal to secondary constriction. The satellite is connected to main body by thin strand. Generally, out of genome of a cell, at least one chromosome possesses a secondary constriction. The satellite is supposed to be concerned with the formation of न्यूक्लियोलस. किसके निर्माण में मदद करता है? केंद्रिका के निर्माण में. ये फैक्ट बताया था मक्का के अंदर मैकलिंगटॉक साइंटिस्ट ने. Next we come to the telomere. The tip of the chromosome is known as telomere. The telomere exhibits unique physiological differentiation from the rest of the chromosome. It prevents the end of the chromosomes from sticking to each other. If the chromosome ends are broken by X-rays, the newly created broken ends undergo fusion. What is the function of telomere? Telomere helps the chromosome in attachment to nuclear envelope. Now, this is all about the structure of the chromosome. Now we are going to see the chemistry of the chromosome. If we see the chemistry of the chromosomes, then you will come to know that the chromosome has the following composition. Number one, DNA histone complex, which is 90%, and the second one is the residual chromosome, which is of 10%. DNA histone complex is again made up of the two parts, DNA, which is 45% of the complex, and basic histone proteins like H2, H2A, H2B, and H4 in 55% of complex. The residual chromosome is made up of three parts, DNA 2 to 3%, RNA 10%, and residual protein 83 to 86%. So this is about the chemistry of the chromosomes. Now next we'll come to the functions of the chromosome. As we all know, the chromosomes are light in the nucleus. So in nucleus they are present and they are made up of DNA. The chromosomes are known as heredity vehicle as they carry heredity unit genes on their surface and transmit the character from parents to the next generation. Chromosomes are having nearly arranged a specialized structure known as gene on their surface. The genes are made up of the genetic material DNA. Hence, together with the enzymes, DNA plays the most important role in the synthesis of RNA, especially messenger RNA, a special kind of proteins, as it continued the necessary message for their synthesis. Thank you.